Welcome in Fundamentals of Human Neuropsychology. In this chapter, we will focus on the central nervous system's hierarchical organization, working from the spinal cord up to the cortex. We will discuss the structure of the cortex, functional theories of brain organization, and we will try to answer one main question. What roles do cerebral hemispheres and subcortical regions play in controlling behavior? An explanation for the apparent disconnect between experiments that support functional localization and those that observe recovery of function is hierarchical organization. Within the brain's functional hierarchy, higher levels provide an animal with more precision and flexibility in behavior. But it's important that we notice that subcortical structures, although lower in the hierarchical organization of the central nervous system, can mediate complex behavior. Because successive steps in evolution have added new levels of brain and behavior complexity, which might have resulted in a hierarchical organization of our nervous system. The brain's considerable capacity for change in response to experience, drugs, hormones, or injury is due to its plasticity, as it is its ability to compensate for loss of function caused by damage. One example of hierarchical organization can be found in examining a complex movement, grooming in the rat. In their study, Kent Berridge and Ian Wishaw established that many levels of the nervous system participate in producing the elements and the syntax, aka the organization, of grooming behavior. Grooming behavior is not produced by one locus in the brain, but rather by many brain areas and levels, reaching from the spinal cord up to the cortex. These successive nervous systems layers do not simply replicate function. Rather, each region adds a different dimension to the behavior. We will now begin with the lowest central nervous system level, the spinal cord, then add structures to see how the corresponding behaviors increase in complexity. Here we have the spinal cord, a center for reflexes. The intact spinal cord can mediate many reflexes, including limb approach to a tactile stimulus and limb withdrawal from a noxious stimulus. The spinal cord also contains the neural circuitry to produce stepping responses and walking, provided that body weight is supported. Now, let's move to the hindbrain, a center for postural support. If the hindbrain and spinal cord remain connected after an injury, but both are disconnected from the rest of the brain, the subject is called a low decerebrate. A low decerebrate animal has difficulty maintaining consciousness because many essential inputs to the brain regions above the injury are now disconnected, presumably leaving the forebrain in the dark, as proposed by Bassett and Penfield in 1922. Cats were generally inactive when undisturbed and showed no effective ability to thermoregulate, which means maintaining normal body temperature. At the same time, they swallowed food placed on their tongues, and therefore they could be fed. If the animals were stimulated lightly in any of a variety of sensory modalities, they moved from their typical reclining position into a crouch. If the stimulation was stronger, they walked, somewhat unsteadily. These stimuli also elicited such typical affective emotional behaviors, including biting, hissing, growling, and tail lashing. Most cranial nerves also have motor nuclei in the hindbrain, whose efferent, outgoing fibers control muscles in the head and neck. Sensory input to the hindbrain is not limited to the cranial nerves. The spinal somatosensory system has access to hindbrain motor system. The same way, the hindbrain has access to spinal motor system. A characteristic behavioral aspect accorded by the hindbrain is a peculiar stiffness called decerebrate rigidity, due to excessive muscle tone. Low decerebrate animals display both types of sleep, slow wave sleep and active sleep, but at different times. Low decerebrate animals also show a sudden collapse, accompanied by the loss of all body tone that lasts from 15 seconds to 12 minutes, which is analogous to active REM sleep. The results of research with low decerebrate animals thus demonstrate that the locus of neural center that produce sleep is in the hindbrain. The behavioral changes seen in low decerebrate animals are paralleled in people who enter a persistent vegetative state, after the type of brainstem damage that essentially separates the lower brainstem from the rest of the brain.